So, dear colleagues, today the topic which I'll be deliberating on is modulating biopharmaceutical properties through crystal engineering. And I put it as an overview because this is only the last decade or so we've started working in this area. So even I'm learning along with you. But yes, I would say this topic, when it was assigned to me, has given me really in-depth knowledge about crystal engineering, and I understand it much better. So thanks once again to Principal K.M. Kundanani College of Pharmacy and the organizing team for giving me this opportunity. And as all of you know, currently I'm at Institute of Chemical Technology, a very premier institute which also has now 2 more campuses, one in Orissa, Bhubaneswar, and another one in Jalna, Aurangabad. So the courses which we offer at the other 2 campuses, that means at Jalna and Bhubaneswar, are integrated courses, and many of them are focused on such multidisciplinary topics as what we'll be discussing today. So let me start by saying, what is crystal engineering? I'm sure Dr. Paradkar would have taken you through plethora of applications, but in simple terms, we can define crystal engineering as exploitation of non-covalent interactions. So these non-covalent interactions can be between molecular or ionic components for rational design of solid state structures. So once the moieties result due to non-covalent interaction, they exhibit very, very interesting properties. So be it electrical, magnetic, or optical properties, they change. And therefore, this arena is catching up. So you can see that crystal engineering is not only important actually in pharmaceuticals, but it is also important in all other fields. So be it then the computers or be it any other electronic items, or for that matter, even in diagnosis. Crystal engineering, this term actually was first introduced by R. Pepinski. And in fact, as I mentioned, it is an understanding of intermolecular interactions in the context of crystal packing. So many questions which arise in mind are whether crystal engineering is just the packing of crystals, whether it starts at the molecular stage, whether it starts only after seeding is done, whether due to the crystal habit, the crystal morphology, etc., properties will change. So there are n number of questions which I feel in the course of this lecture would be clear to you. So obviously, in pharmaceuticals, we are utilizing crystal engineering for designing newer solids which will have desired physical and chemical properties. So when we talk about desired physical and chemical properties, on one hand, we want these structures, our APIs, our excipients, in case we are forming co-crystals, all of them to be stable. And of course, on the other side, we want the desired properties in form of, say, having low melting points so that they can be soluble, they be thermodynamically stable, and yet give us the required solubility, bioavailability, and so on. So definitely, this has considerable practical implications in pharmaceutical industry. So if I have to in a very simplified way explain you, crystal engineering is nothing but our ability to engineer material in context of pharmaceuticals with suitable dissolution characteristics while maintaining its physical and chemical stability. And that's the reason why this is gaining popularity. So does it mean that always crystal engineering is done to get faster dissolution? Answer is no. So you may have to modulate the dissolution characteristics. So can we achieve modulation of release characteristics, be it in form of sustained release, be it in form of increased permeability through skin, be it in form of prolonged residence time on ocular corneal surface. Can all that be done? Answer is yes. So crystal engineering approach can be potentially applied to wide range of crystalline materials. So it offers alternate and potential fruitful method for improving solubility, dissolution rate, bioavailability of poorly soluble drugs. So major application, of course, where it is being used is in enhancing solubility. But as I mentioned, it can also be used to decrease solubility of highly soluble drugs. And that is what is catching up now. You know that more and more moieties which are coming up now are proteins, peptides, macromolecules, which are all nucleic acid, which are water soluble. So can we use crystal engineering to reverse this mode? Answer is yes. And what is it when I say that it offers an alternate and potentially fruitful mode for improving these properties? It's alternative to what? All of you have seen that salt formation is one approach by which you can increase solubility. You've looked at solid dispersions as an approach which could be used to increase solubility. So here what we are trying to say is whatever crystal engineering is done, be it with respect to spherical agglomeration or be it with respect to some polymorphic transitions, be it the solvates, hydrates, etc., all would be aiming at improving physical as well as chemical properties, leading to the best possible desirable pharmaceutical properties. I'm sure all of us here are aware of crystallization process. Even at B Pharm level, no, even if you are teaching pharmaceutics, you have done crystallization. So crystallization is nothing but evolution of crystals from solution. So it could be from solution where you're taking it in atomic state and then it is a bottom-up approach, or it could be melt of the crystalline state from which the crystals start originating. So crystal is nothing but organic compound which is actually ultimate supermolecule. So you'll see in the subsequent slides how the concept totally changed. What we called as crystallization, now people are calling it as supermolecule chemistry. So definitely these crystals assembly would be governed by all the chemical factors as well as geometrical factors, because they are getting built up by individual molecules. So people are using terms like supramolecular synthon. And who doesn't know Professor Desiraju? In case you have not read some of the references and you intend working in the area of crystal engineering, I would suggest, I have put the source at the bottom of the slide. Please go through these references. There's a very good review article given in Journal of American Chemical Society, where this term has been introduced, supramolecular synthon, as I said by him. How does he define it? He says these are structural units within supermolecules which can be formed and assembled or formed or assembled by known conceivable synthetic operations. And what are these operations? These operations involve intermolecular interaction. So if molecules are built by connecting atoms with covalent bonds, these solid state supermolecules, which we call it as crystals, are built by connecting molecules with intermolecular interactions. So we know that there are no covalent bonds. In the beginning, I have said that these are non-covalent interactions. So that is what we are now exploiting in the field of pharmaceuticals. So supramolecular synthon could be homosynthon, wherein the synthon is made up of two similar type of molecules. The name itself suggests, or heterosynthon. Heterosynthon will be synthons made up of two different molecules. That means these molecules will contain two different functional groups which are complementary to each other. So I'm sure now you can really predict where will your co-crystals fall. So when we are talking about co-crystals or newer technology, they are nothing but heterosynthones. Because you are going to react to different functional group containing moieties. So if you have acidic group, you are really trying to see whether I have a co-former which has a amino group and vice versa. Whereas in case of homosynthon, synthons are made up of same type of molecules. So you can see in this slide, I have given structures 1 and 2, which are nothing but homosynthons. So this is for carboxylic acid, this is for amide dimers, but you can have heterosynthones. So can you see just the terminology changes there? From crystals which were known, the hydrates and the solvates which were known, we are now moving to synthons. So supramolecular processes, which are very, very important in crystal growth, one needs to understand. So these are nucleation, and nucleation is nothing but a molecular assembly process where a critical number of molecules are needed to achieve phase change from solution to a crystal form. I'm sure all of you have witnessed this when you were doing crystallization also, in case whatever you are looking at, you're not getting, you can put a seed, right? The technique known as seeding. You can seed a crystal in that solution and suddenly you'll see the crystals growing. So that is the concept of nucleation. And once nucleation has been achieved, crystal growth dominates. So in the crystal growth process, there is evolution of embryonic crystals into an exact crystal size and shape. So it has to stop somewhere. So when it stops, it will have a defined size. It will have a defined shape. And in fact, these properties are now known to affect many pharmaceutical properties. So then the question arises, when molecules interact and form crystals, can we do changes at that level itself so that we can change the moiety in the way we want? Yes, it can be done. And now in today's context, I'm not presenting that case study, but we have started looking again at computational techniques to modulate the crystal properties. You know, when you are doing any kind of MD work, you are having a simulation box where you're putting all the reactants. So when you're putting reactants, if you change the conditions in that simulation box, you get different kind of crystals. And then actually you can see by computer aided drug delivery, which I call it as computational pharmaceutics, whether it will have properties which are required for us. All of you know that crystal is the most stable form. When you have most stable form, it will have least solubility. So when you want stable structure, at the same time you want good solubility, what would you do? We know that hydrates which are there are thermodynamically stable. And hydrates, if you compare it with anhydrous form, I'm sure you know which form has more solubility. It's the anhydrous form which is more soluble. But anhydrous form is also less stable. So can we arrive at a balance between these two so that we can come up with a definite crystal which will be beneficial, is what we need to answer. So yes, pharmaceutical dosage form manufacturers are using all these techniques of controlled crystallization so as to get the right kind of particle size distribution, to get crystals which will be in submicron size ranges. We all know as we are decreasing the size, you know, the surface area is increasing and therefore solubility increases, dissolution rate increases, bioavailability increases. So earlier people used to talk about micronization. Now people are talking about nanonization. I don't know, a day may come and we are talking about piconization also. Already it is happening, right? What is it when we talk about atomic level? What are they? So obviously, this is being done to improve different properties, which are biopharmaceutical properties. We are talking about ultrafine particles. We are talking about converting drugs which are having low solubility to high solubility. We are also talking about some technologies which can keep the drug in the crystalline state. Why? Because generally it is seen when it comes from crystalline to amorphous stage, there will be stability issues. So we are using some kind of charge-based methodologies, tampering or manipulating zeta potentials so that they can remain, the crystals can remain as crystals. So if that is being done, let's see in certain applications, how is it being done? So look at this particular case study where the authors, I have again given the source of references. So those of you who are interested later on can actually go through these DOIs. So nano crystals here were prepared, which are of different shapes. So you can see very clearly that you have C and F, which are rod shaped. You can see spherical ones, which are B and E. And you can also see flaky ones, the plate-like structures, which is represented in A and D. So what are these? Of course, on one side, you have SEM pictures. Other side, you have atomic force microscopy picture. So atomic force microscopy clearly indicates, whether the particles are rod shape or whether they are spherical or whether they have any other plate-like structure or not. So after formulating these with the same drug, the authors have tried to see whether it will have any effect on oral delivery, absorption or not, permeation or not, and what are the transmembrane mechanisms. So this particular paper really very, very clearly says that rod-like nanocrystals exhibited superior performance. Whenever we are talking about mucus permeation, whenever we are talking about in situ intestinal absorption, all of you must be knowing about the ex vivo studies which we do, inverted sac methodology. So those studies have been done and they have actually indicated that if you compare the flake-like, spherical and rod-like nanocrystals, it's the rod-like nanoparticles which interact with the mucus layer well. And it is the rod-like nanoparticles which have given bioavailability also, which is better than spherical and the flaky crystals. So in fact, you can see here that bioavailability was 1.44-fold higher than spherical and 1.8-fold higher than flake-like nanocrystals. Not only this particular study has given account of how this shape can result in endocytosis or exocytosis, transcytosis, so on and so forth. So if we understand this and we know that it's the rod-like nanoparticles which are actually interacting well with mucin, why are all of us trying to get our SEM and TMs which are spherical? So please think about it. If you are getting rod-like structures, can we not use? Here they have used it for oral delivery. Mucin is present even in ophthalmic delivery. So is it that if we formulate rod-like nanocrystals, they will give us better bioavailability when we use ocular route too, or for that matter, vaginal or any other transmucosal route? Point definitely which needs deliberation. Another study wherein they have formed nanocrystals which are embedded in swellable microneedles. And this particular case study is for pulmonary drug delivery, wherein the sinaciguat, this is a drug which is used in case of pulmonary hypertension, is being converted to nanocrystals using high pressure homogenization. And after making the nanocrystals, they have put it in chitosan microparticles. You'll say why? You all know that when we are giving anything as a dry powder inhaler, best particle size for lung delivery is micron range. If you have nanoparticles less than 100, they'll be exhaled. They'll not get deposited in the lung. So make microparticles which have nano in it. The same concept you will see later on in one study we have used for rifampicin, wherein we have formulated NTTP formulation containing rifampicin, isoniazid, and of course the third essential drug, pyrazinamide. But here, what is important is when you have chitosan microspheres, they will be engineered so as to allow sustained drug release because they swell. And nanocrystals which are embedded in these microcrystals, they slowly go on dissolving. So both you are getting retention in pulmonary milieu as well as you are getting the desired release pattern. And this is a graphical abstract of this particular paper wherein they've said that you can get advantages of nanocrystals, which definitely will give you better absorption because once the drug reaches lungs and if it has to reach the finer capillaries within the lung, it has to be nanoform. So that you have achieved using nanocrystals and you're making these nanocrystals respirable, meaning for localization in the lung by making it microparticles. So obviously, these synaceguate nanocrystals are converted, put into matrix of chitosan. TPI is made with the help of lactose, which is again one very good diluent, so that microspheres also get absorbed onto lactose, filled into capsules, and these capsules could be used with rotahellar or cyclohellar or any of the devices so that deposition takes place in lung. And these, when it is in the lung, you know that there is fluid in the lung too. So they are also getting swollen up and releasing the drug in the dissolved form. Another area, we have talked about oral, we talked about pulmonary. Can it be used for transdermal? Yes. Researchers have used these nanocrystals for localizing and sustaining their release at the application site in the skin. Many times you do not want drug to go to the systemic circulation. You want them to be staying in the epidermis because that's their site of action. So can methotrexate nanocrystals be formed which get localized and give sustained release over a period of time? And can that system be better than the oral route where you know there will be a total exposure to all the organs of this particular drug, which obviously you know is an immunomodulator drug. So you don't want that. You want localization. So in this study, microneedle arrays were prepared for intradermal delivery of methotrexate using this particular approach. Simple nanocrystals were prepared of this particular drug, methotrexate, by acid-base neutralization technique, further sonicating it. So once you sonicate, you know, you'll get smaller crystals. Then it is mixed with PVA, PVP hydrogel, and as hydrogel, it is casted on mold. Microneedle molds, one can directly buy actually from market or get it fabricated. Once that is done, you've done the pouring of this hydrogel, apply some positive pressure so that it goes deep inside. You can see what you can see is a brown thing is nanocrystals and blue is a hydrogel matrix. Once casting is done, see that you allow it to dry and then do the studies. So when these studies were done, in fact, it was seen that it was possible to put methotrexate in these microneedles, and these actually acted as sustained release devices. They acted as a depot which released the drug in a very, very sustained fashion. So you can see in comparison to oral, this is what you get as microneedles. But what is very important is whether you're getting the kind of release or the pharmacokinetic profile which are desired. You want lower levels. So you can see that red is what indicates microneedles, whereas your oral one is giving a spike and then it is coming down. So you can maintain levels in a proper desired steady state levels when you're using microneedles. And what do these microneedles contain? Nanocrystals. So I've given you some examples. Another example is, now that we are moving towards, major biotechnological advances, we can't forget to discuss tissue scaffolds. So nanocrystals also have application as bone tissue scaffolds. What people have done here is they have used a natural aerogel material. What is that natural material? It is cellulose nanocrystals. A lot of work is being done on cellulose nanocrystals. And in country like India, this can be a very, very useful resource from waste material. So you can take any waste material, extract cellulose, and then make nanocrystals. So with Dr. Pethe, who is now shifted, of course, to Hyderabad, there is a student who is working on cellulose. And a lot of work in this direction already being done. But definitely rural areas, when we talk about, go to any particular agricultural waste, you can process it to give cellulose nanocrystals. So what did these authors do? They made cellulose nanocrystals, and you can make them porous. So obviously by giving processing, we all know that if you give acid treatment or alkaline treatment, you do get porous gels. I'm sure all of you know this concept that when you're forming a film, say cellulosic film, and you incorporate, say, small molecular weight substances, say like mannitol or urea, and then use this membrane and just pass water through it. So water will take the small molecular weight substances like mannitol, urea, and what would remain will be pores, right? So such porous aerogels can be made. N number of techniques can be used. I've given you just one. So what did these authors do? What they have looked at is whether bone-like cells can be proliferated on these nanocrystals, and whether they are getting the kind of results they wanted. Yes, they did. Not only that, they have not stopped at using only aerogel, which is made up of cellulose. They have sulfated it. Not only sulfated, they have compared two. One is phosphated aerogels, another is sulfated aerogels. And they were compared in terms of strength, long-term stability. And the studies were done actually in rats. So they were implanted into calvarian bone of adult male rats. So it was seen that when these studies were done compared to controls at both 3 and 12 weeks, sulfated aerogels have shown increased bone volume fraction. You can see this. This is control where you can see the black thing, and that is where the bone has been actually, it is not formed. Whereas when you're putting a scaffold, the bone regeneration starts. So sulfated one actually has shown 33% increase in bone volume fraction. At 12 weeks, when you see, it is 50% as compared to control. So what does it say? That means there is evidence of osteoconductivity. So if these authors wanted, meaning if I were in their place, of course, this is done. You know, you can do techniques like CT scan, MRI, etc. But you could also incorporate maybe some fluorescent substance in these aerogels and then see whether how far do they fluoresce, how better they are. So such studies have been done. Not only that, people have looked at effect of crystal form on solubility also. So there's a case study of lamotrigine. In fact, again, this reference is excellent reference if you want to understand how pharmaceutical industry is manipulating and formulating different crystal forms, getting different polymorph and getting different patents, and then moving ahead. So this study has described how the supramolecular synthon approach, which is nothing but crystal forms, can be used to discover novel crystal forms. And then can the relevant preclinical properties be enhanced? Look at this particular slide where they have compared it with different salts and then looked at different crystalline forms. You can see that different crystalline forms are giving different serum concentrations. So this study has been done, say, on lamotrigine, which is anti-epileptic drug, but it will not be restricted to this. What they are trying to do, they'll get a lot of data of different crystal forms, do the kinetic studies, try and establish crystal form properties versus kinetics, feed that data into computers, and then make series of moieties which are similar to lamotrigine and look at QSAR or QSPR also, structure property relationship. So these are the kind of things which are now happening. People have looked at crystal habit also. Earlier I've given you one study. This is another study where sorafenib tosylate has been used, which we know is a BCS class 2 drug. So you can see there are two different crystal forms. These are needle-like. So when you are crystallizing it using different solvents, see if you are taking it out from acetone, this is what you get. And when you crystallize it from N-butanol, you get absolute needle-like habit. This is plate-like, this is needle-like. So whether they have any differences as far as dissolution is concerned, definitely. STB, needle-like, was more polar than STA. So therefore, it has given faster dissolution rate. So if it gives faster dissolution rate, now this is in pH 1.2. Sorry, this is in water and this is in pH 1.2. Whether it will give, many times no, it is seen that they give good in vitro release, but they do not give the desired in vivo. But here you can very clearly see that not only in vitro, in vivo also it has given better pharmacokinetic behavior. So this study is suggesting that if you want to get, many times we are making generic formulations and all we go on doing is adding more and more excipients to get kinetics which are similar to the innovator product. And here the study is demonstrating that just by changing the crystalline habit, you can get modulation in kinetics too. And you'll be surprised because we are dealing with many industrial projects Recently, a company approached us for making an ocular nano suspension. And when we talk about ocular nano suspensions or nano crystals, you know that they are drug crystals, very minimal amount of excipients you are using. Whatever excipient you are using is just to stabilize those nanocrystals. And that's essentially the difference between nanocrystals and nanoparticles, right? In nanoparticles, you will be using some kind of lipid or polymer, either to give you matrix or reservoir, or it could be lipid polymer matrix, or it could be metals. But here you are primarily using API as such. So you hardly have any scope. So if you are not using any of the stabilizers which are permitted to be used, can you not look at this approach? Change the shape and you'll get the desired kinetics. So as I was mentioning, for one, the company working on ocular nanocrystals, we did that. And you will be surprised later when you are looking at Orange Book, Drug Bank and other data, crystal habit was mentioned as one of the essential tests to be done. So not only before exposing it to microfluidics or HPH, but at intermediary stage and later on also. After complete processing is done, we had to make sure that crystalline habit did not change in this case. Because if it changes the solubility characteristics totally. And then accordingly, all the biopharmaceutical properties change. So how many of us actually do SEM at 3 different stages, or TEM if it is required, or for that matter, Raman spectroscopy, which gives you very useful information, needs to be done again. So what are methodologies for controlled crystallization of API? One can use solution enhanced dispersion by using SEDS methodology. One could use rapid expansion of supercritical solution, that's REST technology. It could be supercritical anti-solvent method known as SAS, or it could be gas saturated solutions, suspensions, PGSS process. Now, all these technologies we have been using in our lab, but you need supercritical assembly for the same. Supercritical is a condition where liquid which you are going to use or the gas which you are going to use. For example, in supercritical carbon dioxide technology, we are using carbon dioxide as a gas. We are not using any organic solvent. So you have your API which you are putting it in a chamber, passing supercritical carbon dioxide. So supercritical carbon dioxide will dissolve that moiety, and then through an atomizer, it will come out at a pressure. When it comes out at the pressure, it gives you nano or micro crystals. So that is known as rapid expansion of supercritical solution. In another instance, you can use supercritical gas. It could be any gas. I'm saying carbon dioxide because it's readily available, very easy to use. Supercritical water is also available for your kind information, but requires very high pressures. So you have to use, temperature and pressure which are easy. Like if I would like to use something which happens at 30 to 40 degrees Celsius and doesn't use more than 150 bar pressure. It is safe, right, for people who are using it also. So in case of SAS, you're dissolving your API in a solvent and using supercritical solvent as anti-solvent to throw the crystals out. Same way you can use gas, or you can use certain surfactants along with any of these combinations. So what I have tried to give you by this particular slide is you can control crystallization in the way you wish. So usual methodologies you are aware, all of you are aware, which you've been using. But you can use API along with small amount of any other material which acts as a stabilizer and use these techniques. Co-crystal route, majority of the time again, co-crystal route is being utilized for enhancement of drug solubility and therefore dissolution and bioavailability. Again, co-crystals are attractive alternative to polymorphs. Polymorphs, as you know, are different crystal forms. They are better than salts. Now, salts cannot always get formed. They're better than solvates too. Right? As you know, I'm sure you know the difference between solvates and hydrates. Solvates will have some amount of solvent within the structure, so which remains entrapped. And many times it's seen that just removing that solvent can change the total property, the total solubility characteristic of that moiety. So these are crystalline material which consist of two or more molecular, this is very important, and electrically neutral species, which are again held together by non-covalent forces. And first time, in fact, again, synthon theory for co-crystal and its development was put in 2004. From there, we have moved a long way. 2004 to 2021, we moved a long way. Earlier, whatever references you will see, they are not used pharmaceutically acceptable excipients. In fact, I have evaluated many theses at University of Hyderabad for co-crystal, and everyone knows, lot of work of co-crystals in India was done at University of Hyderabad. In fact, it was in one such meeting that I said, you are doing excellent work, but this work will never be commercialized. And the guide, who was NCL past director, as you know, he was stunned. I said, none of these materials are actually acceptable clinically. So why don't you look at co-formers which are acceptable and then work? And since then, our association is really very, very strong. And he's been able to commercialize some of them. So co-former obviously selected on basis of complementarity. The complementarity has to be there of molecular recognition sites with the API. Oh, this slide I love because this actually tells you if this is pure API, you can represent pure API which is neutral by half moon. And if it is charged API with shaded area like this, these are polymorphs. Hydrates and solvates will have water molecules or solvent molecules like this arranged. Co-crystal can be represented like this, wherein you can have neutral API, but it is getting associated with the co-former. Whereas in salt, when will salts get formed? Only when you have charged API and you have counter ions to match that. So this slide actually puts in perspective what do you mean by polymorphs, by hydrates, solvates, salts, and how your co-crystals are different than all of these. Many people ask me this question at many of the forums. In fact, earlier I used to ask this question at many of the forums, that regulatory, what is their status? So both in US FDA as well as European Medical Agency, it is allowed. So US FDA defines co-crystals as solids which are crystalline materials composed of two or more molecules in the same crystal lattice. EMA also says that they are homogeneous. but it says they are made up of 2 or more components in a definite stoichiometric ratio, where arrangement in the crystal letters is not based on ionic bonds. Regulatory wise, in US FDA, it goes as drug product intermediate, whereas EMA takes it as new active substance. So obviously, that means if you have to file DMF, in EMA you can file, but in US you cannot file, right? Because they are taken as intermediates. So if it is taken as intermediate, you have to propose that it is same as that of parent API. That means if you're taking it internally, after absorption, your API is released. It gets segregated from your co-farmer. That needs to be proved. EMA doesn't say so. So I'm sure this is giving you some insight into if you have to design a moiety, how will you go about? So potential utility of co-crystals obviously is when you don't have ionizable groups. And when you have ionizable, form a salt, go that way, no, it's okay. In fact, most of the industry will do that if they want to enhance pharmaceutical properties. But if it is not having any ionizable groups, see that you form co-crystals. And this is what is termed as solid state engineering. Many advantages it will have, obviously with respect to stability, with respect to bioavailability, and also it will enhance the manufacturing efficiency. There are many methods, solution-based, solid-based. Earlier, co-crystals were formed, earlier ones, whatever references you'll see were all solution-based. Now people are moving to solid-based too. So in solid-based methodology, you can have contact co-crystallization, neat grinding, just like your solid dispersion, ball milling, or liquid assistant, that means wet grinding, or melt crystallization. Again, here I had given a lead to University of Bradford, whether they could use hot melt extrusion as a technique. And the same thing I had told even University of Hyderabad people, and glad to say that it is working. And even one work which we have done, one case study which I'll be presenting, we have formulated using hot melt extrusion, co-crystals. Simple technique, number of steps reduce. Here, the steps of purification are more. So we need to avoid as much as possible. Solvent evaporation, I need not discuss because all of you are aware, right? All of you have done it at some time or the other for crystallization. Same thing will be done here. Only thing is you're taking constituents, dissolving it in a solvent in appropriate stoichiometric ratio, and then evaporating of the solvent. So in fact, many of the people, just to know whether the co-former is correct or not, will do this first. Because by this, you get very pure co-crystal. And if you do single crystal X-ray, you will know that whether it is getting formed or not. XRD is the best method to give you, right, whether your co-crystal has got formed or not. But you have to do single crystal X-ray to know whether it is different than what has been reported or not. So there are many factors which influence solvent evaporation. So obviously you'll look at the solvent type, the ratio which you'll be using, and whether you're using congruent solubility of solvents or not. Many people do that mistake. They do not select a solvent in which both the co-former and the drug API is soluble. As a result of which you may have differential solubility, and at different times they'll come out, they'll precipitate out. So you get a solid mass, but that solid mass is separate API and separate co-former. You don't want that. So use a congruent mix. You should have, for that, you have to understand chemistry well, you have to understand polarity well. And that's why we put so much focus in our undergraduate study. I'm sure Dr. Urmila Joshi will agree with me that how important understanding of polarity of a molecule is. In anti-solvent method, solution of drug and coformer is mixed, and then you are adding the anti-solvent. It can be added using a pump, using a continuous stir tank. So as is shown in this particular slide, right? So you can have impeller. Here you have dissolved your API and coformer in a solvent, and then you are putting an anti-solvent. So anti-solvent has to be pumped in at a specific rate so that you get desired shape and size of these particles. And as I mentioned earlier, instead of using this, we can use supercritical carbon dioxide to pump in as a anti-solvent. It could be, just imagine that you have a peptide moiety which you have solubilized in water. I can use supercritical to take out crystals of those two. What can be simpler than this? So it is need of the hour that we understand chemistry well and take help of our chemistry colleagues to come out with simpler, faster, scalable processes. It's not always necessary to, they give us an insoluble BCS class 2 and 4 moiety and we break our head using either top-down or bottom-up approaches to make them nano. Why can't it be done at the API level itself? Make porous API. Obviously, they'll be more soluble. Cooling crystallization is another technique wherein, as the name suggests, you will be using cooling condensers. So crystal properties with respect to size, purity, morphology, all of them can be controlled. And as can be seen in this particular slide, this is giving a schematic diagram of a continuous oscillatory baffled crystallizer. But is it industrially scalable? If other techniques can be used, we need to avoid this. Because any technique which uses a lot of cooling, a lot of heating is energy intensive. So it becomes costly. So we don't want to add to cost, right? Typically, we want to give cost-effective solution. So if you do not have any other option, this definitely can be used, but is not the one preferred. There could be reaction co-crystallization. This is used when co-crystal components possess different solubilities. What if you cannot arrive at a common solvent? So then you have different stoichiometric ratios of reactants, take them, but control the nucleation and growth of co-crystals. And that can be done by ability of reactants to decrease solubility of co-crystals. So keep it in solution till they get enough time to react. And here again, pharmaceutics can come to rescue of chemistry people or vice versa. Why can't we use techniques like hydrotropy, techniques like microemulsions, wherein reaction can happen over a period of time, bringing reactants which are, even if they are having different solubilities, one is in oil phase, one is in the water phase, can be done. Slurry conversion method. In this method, during the slurry, each component will gradually dissolve. So this forms a complex to promote nucleation and growth. But one needs to really modulate again this process very well. You can use gradient solvent method, just like in HPLC you use gradient HPLC. You can go on adding solvent addition, could be not at once, but in a gradient manner. Contact co-crystallization. This is interaction between API and co-former. Some literature gives it as conformer, but most of the time we use the terminology co-former. So this can occur after soft mixing of raw materials. Possible mechanisms could be, you have two solids, let their vapors diffuse into each other. It could also be eutectic phase formation, could be amorphization, could be anisotropic molecular migration. Anisotropy is a term, is the property will be different when you view it at different velocities or from different perspectives. It could be any property. So higher humidity, higher temperature, small particle sizes of raw materials all actually can facilitate co-crystal formation. So you may be thinking, whether when you have high humidity, if you have hygroscopic material, whether they'll form co-crystals. Yes, they will form, but then it is your headache. How do you crystallize out? So couple the techniques together. You can use anti-solvent technique along with this. Neat grinding, where you're using, I've already told you, using ball milling or any other vibratory mill. We are using at ICT planetary ball mill. Same way, when for liquid assisted grinding, we've been using planetary ball mill. Of course, ball mill also be used, but planetary is much more efficient. And in planetary, you can use zirconium balls. Now, zirconium is one metal, basically very, very hard material, just like diamond, but doesn't give residues like metal will do in case of ball mill. And that's the advantage which you get. Melt crystallization is a green approach, as I told you, Hot melt extrusion, similar to melt crystallization. So solvents are not involved at all. So you can use your API along with co-former, extrude it out. Of course, one drawback of this technique is that both the API as well as co-former have to be heat stable. So that you need to check. Otherwise, total change may happen. So if you are working in these ways, definitely you can influence the biological voyage. So depending on route of administration, depending on how the co-former or co-crystals are getting distributed in the body, depending on the acidity, the pHs, the enzymatic processes, everything will get affected by these, as well as these can affect your co-crystals or crystal engineers or synthons, whatever you might want to tell it. So all of these properties are going to get influenced. So any time you are making new drug delivery systems, your objectives are that you get improved drug release. Specifically, in vivo kinetics need to be improved. Why I've said improve? Because sometimes you may want faster Tmax, higher Cmax, but sometimes you might want less Cmax, but over a prolonged time. So that should be possible by any NDDS. You should get enhanced drug absorption, could be site directed, because now we are talking about targeted delivery. So I give you one example, right, where rod-like particles were interacting well. So obviously they'll get retained well. So same way, you can look at crystal engineering aspects and see. We have, some lipid-based system. You will be shocked when we are giving it orally. 90% of it is taken by the lung route, meaning by the lungs. So that is site-specific drug distribution. And why is that happening? Is it the chemistry of my nanoparticles? No. It is the shape of those nanoparticles which is taking it to that site. So those papers already actually are there, published papers. Physicochemical properties which you need to gauge and pay attention to is particle size, surface charge. Surface charge plays a very important role with respect to stability. So you need to look at that. Obviously, with respect to particle size, the solubility will go on changing. You need to decide whether you'll add functional excipients or without functional excipients, you'll get the desired release profile. As for example, I told you, if you're making co-crystals, but you want them to be prolonged release, unlike what others are using it for, you want them to be prolonged release. So can you use polymeric moieties which have functional groups which you want to interact with your API? Definitely it can be done. If you can entrap them and make reservoir systems, why can't you make them react? Surface modifications also can be done, and you'll get active targeting ligands. So you can make nano crystals of which surface has been modified. So can nanocrystals be modulated in such a way that it has, say, transferrin on it or lectin on it? Who says it cannot be done? In fact, in our dictionary, impossible does not exist. So it shouldn't exist in anyone's dictionary, actually. Co-crystal, what influence it has on hygroscopicity? One example is here. We all know, and I've already told you, that anhydrates show higher solubility than hydrate. Why? Because hydrate actually is the thermodynamically stable form. So always stable form have lower solubility. So in this case, you can see co-crystal of xylitol were formed. This is the form A of xylitol, which interacts with nicotinamide, and it also reacts with isonicotinamide. So this particular author, Chen et al., have prepared co-crystals. You can see that in high relative humidity condition, this form A changes. Look at the structure here, what form it takes, right? So form H gets formed. So results actually have indicated that when you form A reacts with nicotinamide or isonicotinamide, co-crystals is formed, and this co-crystal has superior phase stability against moisture. This particular drug is used for asthma. So in this case, obviously, if you have to show that co-crystals are formed, you can see the powdered XRD patterns. So you are recording it for form A. So A, B, C in this particular diffractogram are indicating form A. Then you have with nicotinamide D and E with nicotinamide. Then you have with isonicotinamide, that is F and G, and then there is form H. Very clearly you can see if this is the original moiety, form A. This is original. After 1 week, same peaks. After 4 weeks, one peak starts appearing, which is shown by arrow. But when you are making co-crystals, see, that peak is not there. But when it gets converted again to form H, the peak starts. So this indicates that if you can make co-crystals, definitely you are resolving stability issue also. So in this particular study, they have shown anhydrous xylitol is giving higher IDR than monohydrate xylitol, or you can say that monohydrate xylitol, which is there, is less soluble as compared to its anhydrous form. And of course, co-crystals have given better intrinsic dissolution rate. So what you are getting is advantage of both. This was not stable, so you have stabilized. this was not soluble, so you have solubilized. So such research needs to be undertaken to come out with co-crystals which are physically stable yet more soluble. Co-crystallization also has shown its potential to improve in vivo performance for poorly water-soluble drug, and in this case, the case studies of ketoconazole. And ketoconazole, all of you know, is antifungal drug, old drug, but see, been revived, giving new form. What did they do? They crystallized 1:1 co-crystal of ketoconazole and PABA. And that has given them 10-fold higher aqueous solubility. So is water solubility the only thing? See, you know, there are drugs which have very good permeability. So all you have to do is make them soluble so that they can give you the gradient which is required. So by doing that, it has given them 6.7-fold oral bioavailability improvement too. And obviously, you know that PABA is not toxic. So they've used PABA as a co-former here. So this gives the results. This is how ketoconazole behaves. It's almost parallel to the time axis. Whereas when you are having ketoconazole with PABA co-crystal, see the kind of increase it gives. So they form co-crystals, confirmed its identity, confirmed that it gives consistent crystallinity in bulk also, shown that it is stable on storage, shown it is more soluble. Not only that, can you see this particular kinetic profile where very, very high Cmax is reached. Not only that, the level also is maintained for longer period of time as compared to only ketoconazole. So obviously, you know what is important in case of antibiotics or antifungal? Not only the effect, onset is important, but post-antibiotic or post-antifungal effect is more important. Meaning, once you give, for how long it can maintain that effect. So such formulations can give good post-antifungal effect too. This is by, of course, they have looked at by oral route, but who stops you to look by topical route? You know, fungal infections are localized, most of them. So can't you give it in such a way that, and use co-formers in such a way which give you localization, give you depot effect, but make the ketoconazole or any other azole for that matter more available? Think. So another example is of sustained release where drug co-crystals have formed. That is the area which is picking up now. And you'll see later on one case example even from our lab. What they have done? Isoniazide, which is a highly water soluble drug. They have formed co-crystal with curcumin. You know, curcumin is not soluble. So by forming isoniazide curcumin co-crystal, they have attained sustained release. Look at this. This is for the co-crystal. The blue thing is for the co-crystal. Okay. And that for rifampicin and isoniazide generally is taken, you know, together. I told you rifampicin, isoniazide, pyrazinamide, like 3-drug combination is the gold standard. So therefore you can see here rifampicin also, but since I talked about isoniazide, for isoniazide also very clearly, you can look at what would be the release at different pHs. And this way what they have actually shown is not only you can sustain the release, you can see the releases for 48 hours, but by forming these co-crystals, stability is also achieved. Many of these drugs are not stable in GI milieu. Now, if you read, Professor Shishu's paper, which have been published from LMCP and later on from PERT Center, you'll realize that most of the drug goes waste. You know how high dose of the drugs have to be given. Why high dose? Because most of it is wasted. So by doing this and releasing only small fractions at a time, can we increase stability? Again, answer is yes. So therapeutic effect of drug substance, we know is actually influenced by physicochemical property. You can actually restrict the potential if you have BCS class 2 and 4 drugs. But if you make co-crystals, obviously you can enhance this particular effect. And you've seen it in many of these studies. So now we'll move on and look at some of the work which we have done at ICT using crystal engineering approaches. And the first one which I would like to tell you is novel nanocarriers of curcumin for Alzheimer's disease management via intranasal route. Now keep in mind, we are using here a dual synergistic approach. Why I'm saying dual? Because first I have made co-crystals and then I have put co-crystals in my cell. If I directly want to put something in my cell, there is limitation of how much I can load. But by increasing the solubility of curcumin by forming a co-crystal and then loading it in my cell, I'm getting all the benefits. Now conformer A, which we have selected, also has antioxidant potential. And for Alzheimer's, antioxidants are very important. So it gives good stability, better biopharmaceutical properties. And when we make my cells and give it through intranasal route, very, very high amounts of my cells go to the brain side. And because co-crystal is encapsulated in that carrier, over a period of time, we'll get control release. So such are the technologies which are coming forth. In fact, this paper I have published in a very special issue in Journal of Pharmaceutical Sciences. It was a call in honor of one of the scientists, and we could contribute this. In fact, without any corrections, it got accepted. Of course, what did we do in this case? We started with computational study. So we looked at curcumin co-former interaction. What you want when you're looking at such particular studies, we look at what is energy, right? Energy is what directs everything. So energy of complexation has to be high, in fact, has to have a positive value. If you get such a value, that means it is a stable complex. So first step was we actually screened many co-formers which had the potential antioxidant action. And this particular one, which again, because it is published, but we have not revealed which is a co-former A, still looking at whether we get industrial partners. But this gives you idea as to how did we go about. And you can very well think, if you know what is a curcumin structure, what are the functional groups, anyone the common sense will be able to actually guess what would be the co-former. So it gave us that at 1:1 molar ratio, there's a very good complexation happening. Having done that, we utilized, this is a planetary ball mill, which we utilized. All we had to do is put it in the jar, put it along with zirconium balls, and then later, once we get the powder material, sieve it through 60 mesh and fill it in capsules. Simple process. So we have used grinding process to make co-crystals. After making these, obviously, We had to optimize it. So we have utilized design of experiments. So looked at independent variables like what could be the RPM, what could be the process time, studied it at 3 different levels, and then came out with a design space. So very clearly you can see that predicted values are really matching with the actual values. So then we went ahead, we got yield also, which was quite good. 97% yield is really very good. It's not moving ahead. Just give me a second. I'm sure you know that in such cases you are using, because you are looking at 3 levels and we had 2 independent variables. The factorial design which will be used will be 3:2. That means there will be 9 such batches which you will get, and then you will work on all those batches, plot the data. We have used design expert here, and then we have gone ahead and looked at the control strategy. I'm very sorry, it's not moving forward. Can I reshare if you don't mind?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes. Dr. Mrinal, could you tell me how much time do I have so that I can go accordingly?
Ma'am, 15 minutes more.
Okay.
Your session is till 4:15, including question and answer.
Okay. Still getting loaded.
Some network issue.
Is the slide visible or I have to reshare?
Ma'am, we can see your this factorial design slide, design of experiment.
Oh, it is still
Controls. Yes, still the same, yeah.
now.
Yes, now it is visible.
Which is visible now?
Ma'am, it was there, then suddenly it got disappeared.
Is it
Can you
again?
share it? Yes, yes.
Oh my
It
God.
was, yeah.
Hmm.
Yeah, ma'am. The T micellar nanocarrier characterization.
Again, it has gone to slide number 1.
Yes,
right?
yes, ma'am. Yeah. So started from the beginning.
Done?
Yes, yes, ma'am.
Sorry for that. Dear colleagues, sometimes there are technical glitches. So as I mentioned that we formed co-crystals and then we loaded them into micellar nanocarriers because we wanted to take dual advantages. So they were duly characterized. And then what was important for us was to look at what happens when in vivo studies are done. Oh, God. And directly I'm moving to in vivo brain biodistribution studies, wherein you can see that there are different groups. But what lines you see in purple are the lines which you have got with co-crystal micelles. Of course, we knew that we've used T ligand. So that means we had, in this case, micelle formation, wherein we have synthesized using one-step reaction amino acid based T ligand which self assembles us to give T micelles. But when you have co-crystals which are entrapped in T micelles, they are giving us much better biodistribution as compared to even micelles. I hope you're getting it. We made T ligand which was self-assembling to give T micelles. T stands for targeted. So that had curcumin in it. Now in these micelles, what we have entrapped is curcumin, which has co-former to form co-crystals. So that has given us much, much better brain biodistribution. And you can see when I plot cerebellum or I plot it in cerebrum, everywhere prolonged release is seen with co-crystals which are there in my cells. So this proved our hypothesis. Of course, we did stability studies also. Room temperature stability studies, you can see for 12 months. And we've done accelerated stability studies as per ICH guidelines also. And they were stable. So curcumin co-crystal micellar nasal spray is stable. And in fact, by doing this, we have proven that we can do a very systematic study, identify antioxidant co-former, apply DOE, use planetary ball mill to get co-crystals in one step, make micelles which have size less than 25 nanometer, make a nasal spray, and in all this, now we have utilized colorless curcumin. So we have strategic combination approach of curcumin co-crystal micelles. As I mentioned, it's already published in Journal of Pharmaceutical Sciences, and maybe you can go through that. By doing this, we read so much that with one of our undergraduate students, we have published Crystal Engineering, Upcoming Paradigm for Efficacious Pulmonary Drug Delivery. There's a book chapter. Not only that, in Pharmaceutical Biz, your tabloid, we pose this question, whether they are the next big thing. And this is in year 2013. So you can see that how big now they have become. Next is novel formulation for therapy of tuberculosis. And I'm sure all of you know about tuberculosis, so I will not take much time here. But yes, it's a very, very contagious disease. One can get pulmonary tuberculosis, which resides, localizes only in the lungs, or it could be extrapulmonary. So extrapulmonary could be either brain or could be bones. It could be many of the organs. Many times it does go undiagnosed. Many times if you have low grade fever and you ignore, go on taking paracetamol, it's better that you get tested for this. So tuberculosis, the top infectious killer in the world, we know. The drawbacks of current therapy are that you need to take it for too long. And in fact, most of the drug, as I mentioned, which are used as first-line therapy are high drug doses. Most of it is going actually dumped in our body, so there are severe side effects. And it is because of these side effects that people discontinue therapy. And the moment they discontinue, you know that resistance will get developed. And that's the reason why we are getting now multi-drug resistant TB cases. So we thought, can we make these as nano? So when we are making our API in a nano form, obviously dose will reduce. Our prior experience says that dose can reduce by 1/3 to 1/50th. So if that is it, why can't we reduce dose? Second thing we thought is, why give it orally? Can we give it in case of pulmonary TB at least by pulmonary route? So can we get that benefit? And rifampicin, as you know, is the culprit. So in this case, what we have done is we have looked at making rifampicin nanocrystals, which will be given as DPI. In fact, this particular formulation has all three drugs, but the case study which I'll show will be for rifampicin, because other two are nanoparticles. We wanted release in a prolonged fashion, so they are nanoparticles, but we wanted to give rifampicin in the faster mode, so it is a nanocrystal. So very systematically, we started with making rifampicin nanocrystals, looked at solubility, of course, looked at contact angle measurements also. Always we do this side by side. So obviously, when we are looking at saturation solubility and you are looking at nanocrystals, you do not want drug to be highly solubilized. So what people do the mistake, SLS is showing solubility, let me pick up this for my nano formulation. Don't do that. In fact, you have to pick up surfactant or a stabilizer in which it shows least solubility and of course lower size. And same way, sorry, going back one slide, same way contact angle measurement is important. You should have better wettability. If you have better wettability, processing is easier. So for that, there should be lower contact angle. Should not be very high contact angle. So we found in this case Poloxamer 407 to be doing the job better. So obviously it was characterized by various means. Pre-formulation studies were done. Using DOE approach, we made rifampicin nanocrystals at drug to surfactant ratio of 1:1. Went in a very, very systematic mode. Looked at media milling as well as microfluidization as our approach. We wanted to see which will be better. So we have used high pressure homogenization as well as microfluidization. So in a very systematic manner, we have gone. In fact, this paper is also now published. So comparison of ball mill as well as microfluidizer. And you can see that pure rifampicin looks like this. But when you're converting it in ball mill, its morphology changes. In fact, when you process it through microfluidizer, it becomes very, very smooth. So this is from rod-like structure, cubes, from cube-like structure, in fact, smooth structures. So obviously, its dissolution also changes. So will its saturation solubility. So after establishing this, we have gone and looked at dissolution in lung fluid too. So you can see that in vitro dissolution in simulated lung fluid with rifampicin, which is ball milled, and with rifampicin, which is microfluidized, is much, much higher than the physical mixture or drug alone. But was this it? What we wanted to do is efficacy. So we did REMA. This is with M. tuberculosis. In fact, very virulent pathogenic organism. So you can see that the results do indicate that after this particular concentration, there is a change when we have our rifampicin formulation. So drug here is also in solution form, because for all these assays, you have to take drug in solution form, which you are dissolving it in solvents. But you see the color changing only here. Here, the color changes much earlier. That means its MIC is much less. Or in other words, you can say that its efficacy is much more. So we looked at in vitro cytotoxicity also by MTT-assay in raw cell line. Of course, that's a cell line for lungs. So it shouldn't be that it is efficacious but kills my normal cells. So that was not happening. And not only that, look at this graph. When you give it oral, Cmax is coming later. And of course, levels are there for a long period of time. But when you're giving it in a site-specific fashion, lung deposition is much more, much higher and much more. This is with nano crystals. So that means if I want to match this, someone may say, you don't need such higher levels. If this is giving me efficacy, I can now lower the dose and get still faster onset of action. Onset of action is much faster here as compared to oral route. So I think this has given you enough idea about what is the advantage of rifampicin nanocrystals, which were actually formulated using both the techniques. Size here is 600 to 900 nanometers. We've done extensive study. I'm only presenting the part which is relevant because this has to be formulated as DPI. So you have to do Anderson pipette method. You have to do all the cascade impaction, all those studies, and then select a formulation, administer it to animals. All that is really intricate. Another one which is my favorite always, which I'm not leaving this field, and this study has been done by undergraduate students. Novel co-crystals of anti-malarial drug combination. That is drug co-crystal. So you can see the drug when it is taken in a particular solvent, how does it look? Drug in IPA water, how does it look? We mix them both. So when we mix them both and then dry it, we've done two methods. One is the same simple rotary evaporation, because you just need to evaporate the solvent, right? Other is we have also used RES. I told you we have supercritical setup. So rapid expansion of supercritical solvent technique. So what we get is a powder. So just by looking at actually the color, we knew that co-crystal has got formed. But this was given as a BTech project to a student. So obviously he did all the studies. So we characterize it by IR. We looked at DAC pattern. So obviously you should get a new peak, if it is a co-crystal. You should see fingerprinting region differences you will see in the FTIR also. We did NMR. We proved by mass that co-former, these two drugs actually, each one acting as a co-former for other, they are interacting. So by mass also that was proved. Then we went on to powder XRD, and very clearly you can see that there were different peaks. In XRD, crystallinity was about 33%. So that was crystalline material. We purified it. Not only that, very clearly you can see this is a co-crystal powder XRD, which is different than the pure drugs. We did single crystal XRD too. And when we did single crystal XRD, you can see the images. at different magnifications. So they were much different than what they were original. That also proved that we have a co-crystal here. So now, in vivo of this can be done. And what we believe is that when this will be taken internally, it will segregate as two different moieties and exhibit the action, which is a desired action. So we have seen that in this case, there was about 42% dose reduction. We have used a WHO approved combination. We have used a green technology, no solvent there. A very viable means of bioavailability improvement. In fact, here extensively we have again used computational method. Because we do not nowadays start, and waste our efforts. We start with computational means. And it has scale-up potential too. So that means we are ready to go now to next level. Last studies of atavacone nano suspension. Again, in this case, we have optimized the formulation, and you can see that initially we had needle-like crystals. We have converted them to cube-like. Extensive characterization done, and we have gone up to in vivo stage, wherein we have shown that it matches actually in vivo with Mepron, which is a marketed product. But to match it, you will be surprised, we had to lower the dose 50 times. Okay, so Mepron is not a nano formulation. So if I want a generic product, I can lower the dose. If I want to go NDA way, I can go with this dose, lower dose, and see whether I get any additional benefits without the toxicity. That's the next step. So again, in this case, we have, by utilizing the co-crystal approach, proved either co-crystal or crystal engineering or nanocrystals or microcrystals, we have proved that this technology definitely is here to stay. And just for your information, since I'm working a lot with devices also, we are coating nanocrystals on devices, and that work already has gone to pig level, being evaluated in pigs. So these are some of the technology transfers which we have done. Our collaborators are many, and we thrive just because we have good industrial partners and government grants. And these are some of the books which our research family, I would say, have contributed. And our lab is equipped to do any kind of such work. We also have a cell culture lab. And with that, I would like to conclude. We follow this motto of Swami Vivekananda ji, which is focus. Though we have diverse area, whomever the topic is assigned to focuses. And this is our happy VBP family, who's been instrumental in collecting, collating this data and helping me put it together. Of course, we have done a lot of work in lab, and therefore that data was available, but some of it you have seen is from literature. So thanks to family members for helping me with this. With that, I would like to stop this presentation and take any questions if you might have. Thank you, and thanks once again to KMKCP for giving me this opportunity.
Thank you so much, ma'am. It was very informative and valuable session for all of us. I would request Rajni, Dr. Rajni ma'am, to please thank you. Hello, hello, Rajni ma'am. Yeah, ma'am.
Madam, thank you so much for such a detailed discussion on crystal engineering, starting from basic properties, process, applications, supramolecular synthon theory, then various clinical applications along with discussion on the case studies. Madam, I remember my BPharm lecture. I always miss your lecture. Thank you so much.
Congratulations.
Yeah.
Vandana, heartiest congratulations on behalf of your alma mater. Everybody is congratulating you.
You know, I think this is the first forum where I'm hearing this. I've yet to see my mobile, not opened it. So thank you so much for breaking this news to me. I knew that I had applied for it, but getting this in his name, you know, I always wanted to meet this great personality. And even when I visited Rashtrapati Bhavan for Gyati Awardee, I was talking to his photograph. I said, in this lifetime, I didn't get a chance, but getting this fellowship in his name is a real good thing which has happened to me, let me tell you. And this motivates me further to do my best.
Hearty congratulations, madam. And on behalf of KMK, we all congratulate for your honors which you have received. Thank you.
Thank you so much. I was answering Dr. Harsha's question. So as I mentioned, you know, you need to understand the chemistry well. Keep in mind that there is no covalent interaction happening. So obviously, it is the hydrogen bonds which will play a very important role. So typically, as I mentioned, in some of the cases, no, when you have amino groups, you like to interact it with acidic groups. So you have bonds which are formed so that in the body, when they go and they break, if that doesn't happen, your moiety will not be released, right? So it's very important that you have such bond formation, and amide bond is one the simplest one to go ahead with.
We have our next question from Vandana Panda ma'am. She's asking which drugs you have used in multi-drug co-crystal which you have prepared.
So it was anti-malarial, right? Already you know that etavacone and proguanil is a proven combination. It's a WHO combination. And this particular moiety not only goes in for malaria, it's used for many of the other infections also, you know, parasitic infections also. In fact, it's also given us prophylactic to for anti-malarial, those people who are coming to our country or in Africa, all travelers are given this. So we have combined the same drugs which otherwise will be done the formulation way, we have done it in the co-crystal way. I hope you got the answer. Etavacone and proguanil hydrochloride.
Yeah, ma'am. Any other question from anyone or someone wants to interact with madam can raise your hand.
Madam, I have one. If there is any dose limitations and molecular size limitations.
Your voice is not very clear, you say?
Rajni ma'am, can you speak bit louder?
Yeah, madam, is there is any dose limitation, like, you know, the size, dose of the drug we have to take into consideration and the size of the drug?
Right. So I feel very intelligent engineering needs to be done there. And that's where we take, you know, help of computational tools. So there is no limitation as such. Absolutely no limitation. But in fact, you know, what could be a problem is, now suppose, especially with respect to drug co-crystals, it may be that dose-wise, clinicians are prescribing it in 1:4 ratio. But co-crystal wise, you are getting it in stoichiometry 1:1. Then what would you do? So that could be an issue. So one needs to very critically evaluate dose response curve for such drug co-crystals. But for co-crystals as such, there is no limitation. At least I have not faced so far.
Okay, ma'am. Yes, thank you, ma'am. Yeah.
Anyone? Any queries? Yeah, Vandana ma'am, Vandana Panda ma'am, you want to ask something?
Yes. Congratulations, Vandana. Further to the same multi-drug crystal, co-crystal. Now, is it easy to do that? To put multiple drugs together? You've done it for anti-malarials, but I wanted to know about your experience. That is it an easy process?
It is, see, once, no, Dr. Panda, if you understand the chemistry well, right, and if you have two drugs belonging to the same class, which increase each other's solubility, okay, definitely it can be done. It's an easy task. It's one-step task. It's not difficult at all. Solvent evaporation method is easiest, right? You've been using it. All you have to do is look at a solvent in which both the drugs are soluble, and then just evaporate off the solvent. But it's that easy.
Okay, thank you. I thought that would be difficult to put two drugs together, but
only thing is what you have written in your question. Not all drugs can be combined. That's absolutely correct. You have to have moieties which can interact without forming a covalent bond, right? So that identification is important.
Thank you.
Yeah, ma'am, we have one more question from Anuja Bhosle. She is asking, what is the criteria for multi-drug selection for co-crystal formation?
So you want complementary efficacy, isn't it? Now, for example, you know that if I have a skeletal muscle relaxant and I give it with antipyretic, it would be useful. So clinical perspective also you should have. Malaria is all, meaning generally, you know, with fever, it is associated with fever. So can I have a combination of anti-malarial drug with an antipyretic so that both the things can be tackled? So there is a clinical perspective and there's a chemical perspective. So I think if you look at that, it would be easy to go. In chat box, I can also see another question, discuss about patent implications of co-crystals or
polymorph.
So earlier, you know, polymorphs, even US FDA was allowing. So what people used to do is actually file a patent for one crystal form, then second, then third, and so on. But now Indian Patent Office is not allowing at all. So, but co-crystals, as I said, since regulatory norms are in place and it is considered as an intermediate, it can be filed. So co-crystals can be filed, no problem.
Ma'am, related to that, one more question is that is how to approach for patent-oriented formulation.
That would depend on n number of factors, right, Anuja? Yeah. Whether which route you want to go ahead with. Starts with that. Second, which methodology you would want to make. So you can take patent for making a co-crystal, that means a process, or it could be like what we did. We made co-crystals and finally we have put it in micelle and then patented it. So it could be a formulation patent which also covers process. So depends on n number of things. What is the drug in question? Whether for that particular drug already someone has taken co-crystal patent, which is a broader patent. If already co-crystal process patent has been taken and you were thinking also of the same process, why don't you go for formulation patent? So you can still take drug-specific formulation patent. Still can be taken. With one our industrial partner, we have taken for psoriasis drug-specific patent. Can be done.
Ma'am, Harsha ma'am has asked one more question. Can atovaquone co-crystal be made for increasing its solubility and bioavailability?
Yes. So we have already done that. I'm not shown you, but already that has been done. Definitely it should be done for such drugs. So any drug which is in BCS 2 and 4, solubility and bioavailability can be enhanced using this approach.
Hello.
Ma'am, doctor.
Hello.
Hello.
Madam, VS College of Pharmacy. Hello. Am I audible?
Yes, yes, you are audible.
Yes, Dr.
Yes,
Rajeshri.
yes. Heartiest congratulations, madam, first of all, and a very nice informative session as usual. I have one basic doubt, that is, after we do nanocrystallization, the final nanocrystals which we get, do they have any stability issues? And if so, what precautions should be taken to maintain their stability?
So thanks. Is that Rajshri? I can't see you, but I think so from your voice.
Yes, yes.
Right. So thank you so much. First and second is yes, nanocrystals. As I mentioned, when you're making, you can use small amount of polymeric stabilizers. So these polymeric stabilizers can actually coat the crystals and give them stability. And many times it's not that nanocrystals alone you are filling in a capsule, right? You can take help of other excipients which go as usual in a tablet or in a capsule. So you can add a filler. So once I had faced this issue wherein when we added filler, you are typically like SMED formulations, you are absorbing them and then converting them to free flow. But nanocrystals which you get are in solid form. And crystalline materials more or less are much more stable. Now stability could be the drug's inherent instability issue. For example, drugs might be undergoing oxidation. So then along with that filler, you can actually fill antioxidant and take it ahead. So once we had to do that also. Thanks for asking that question. Important attribute, yeah.
Yeah, thank you so much, madam. Another thing, I just wanted to know the microfluidics, that is the one which is available, the same instrument you are using, the microfluidics?
Yes, yes.
As a brand name microfluidics, right?
Same. We have a microfluidics.
Yes.
Yeah.
Thank you so much, madam. Missing you.
Hello.
Thank you.
Hello. Hello. Good evening, madam. Am I audible, madam?
Yes, you are audible.
Okay. Heartiest congratulations, respects, madam. And it was very nice session. Valuable insight on co-crystals. And it would be really useful for we student, madam, in future, nearby future. So one question, madam, from my side. If we go for drug co-crystalline, is there any change in the pharmacological activity of the drug when we go for drug co-crystalline?
Another aspect, I think that's Popat talking, right? Popat Kumbar? Yeah, so such studies are not yet out because this is a very nascent field. But a very, very important question which has been raised by you, that you need to see whether such combinations can give some other activity, just like, we have R form, S form, and then RS giving different activity. You should establish whether you're getting the inherent activity without the toxicity, or whether this particular drug combination is giving any additional activity. So that needs to be established. At least till date, I have not come across any such paper. But if I do come across, definitely I'll pass it on to you.
Yes, ma'am. Yes. Thank you. Thank you so much, ma'am.
Great thought process. I'm happy that I'm getting many, ideas.
It's inspiration from you, madam, only.
Thank you.
Thank you, madam. Thank you.
Ma'am, one last question by Dr. Vrish Dhwaj. He is asking, what is the validation method for ensuring efficacy of drug which is given by intranasal route?
What I do not know whether I understand the question properly.
Yeah, he's asking the validation method.
I can read that. So majorly, proof of concept is in having the efficacy, right? So finally, when we have done, for many of the intranasal formulation, what we do is we either radio label the drug moiety, or we say put fluorescent dye in the our nano carrier, and then we administer it by nasal route. So it is never that 100% which you have sent by nasal route goes to the brain, if that is what you are looking at. Okay, So part of it goes to the brain directly through olfactory, bulb, through axonal transport or through trigeminal nerve. But most of it might go through paracellular route of the nasal mucosa into the blood, and then from blood again go to the brain also. Okay. So I would say, meaning in one way of answering that would be to look at whatever you had proposed. Suppose I'm working on, say, glioblastoma, and I want that my drug temozolomide should go by intranasal route in much higher amounts to brain. So can in animal studies, I can plan in such a way that I radiolabel my drug, put it in my cells, administer it, and then after the study is done, take the sections of the brain, estimate what is the radioactivity count in each part of the brain. That will give me an idea where in brain it is going and in what amount it is going. It's not that you have to do radio labeling. You can also do biodistribution studies, wherein you take the section, you mince the tissue, right, each part, and then see how much has gone there. So obviously, I mean, simple in vitro tools are not going to tell you or validate that it will have efficacy. You need to do in vivo studies finally.
Thank you so much, ma'am. It looks like we have covered all our questions. If still any queries are there, we will mail you. We have one question from Harsha Katpalia from VS College of Pharmacy. She is asking, what is the criteria for selection of co-formers?
So for that, Dr.